the human body develops, replaces old cells with new cells and repairs itself with the help of cells that constantly multiplies by dividing. This process is known as cell cycle and is of two major phases. The preparatory phase known as the interphase and the cell division phase known as the M phase, where the letter M stands for either mitosis or meiosis. Before we go deeper, let's look at the difference between the mitosis and meiosis. The mitosis occurs in somatic cells, which includes the skin cells and every other cell except the reproductive cells, while meiosis occurs exclusively in the reproductive cells with respect to the gender of the host, to produce haploid sperm cells as in the case of a male and haploid egg cells as in the case of a female. Now let's look at the stages involved in mitosis. The first stage is the interphase, where the cell grows and is preparing for division. Then prophase, when identical copies of DNA molecules organize into chromatic pairs. These pairs of chromosomes connect to each other at the centromere, and the nuclear membrane disappears. After prophase comes the metaphase. In this stage, the chromatids align themselves along the cell midline, spindle fibers attached to the centromere. In the next stage, called anaphase, the chromatids are pulled apart by the spindle fibers. After that comes the telophase. A nuclear membrane forms around the two exact sets of DNA. Finally, the cytokinesis, where the cell totally divides, forming two identical daughter cells. On the other hand of cell division is meiosis. In meiosis, the cell undergoes two very important stages, simply known as stage 1 and stage 2. Each stage has several phases. In stage 1, very similar to mitosis, the cell goes through interphase 1, where it is growing and getting ready for division. Prophase 1, when copies of DNA molecules organize into chromatic pairs and connect with each other. Then metaphase 1, where the chromatids align themselves at the center and spindle fibers attach to the centromere. Next is the anaphase 1, where homologous chromosomes separate and move to opposite poles. Now in stage 2, there's prophase 2, where the chromosomes condense again. Metaphase 2, where the chromosomes line up at the center again. Anaphase 2, where sister chromatids separate and move to the opposite poles. Telophase 2, where a nuclear membrane reforms. And then finally, the cytokinesis, where the cell totally separates and forms four daughter cells.